Technical Veterinary Learning Channel. Honored by Prof.Ahmed Mamdouh El-Sherif, Professor of Epidemiology and Infectious Diseases, the ex-head of Department of Veterinary Medicine, the ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University, presents the West Nile Encephalitis. It is an important disease with nervous manifestation of family Equidae. West Nile Encephalitis disease. It is a potentially an infectious disease of family Equidae and a variety of mammals with human zoonotic impact. West Nile Encephalitis. It is an infectious disease characterized by fever, fatal encephalomyelitis, as mean inflammation of the brain and the spinal cord, and usually ended by death. History of the disease. West Nile fever was first recognized when isolated from a feverish woman in the West Nile district in Uganda in 1937. Since this disease has been found in humans, birds, and other vertebrate in Africa, Eastern Europe, West Asia, and the Middle East. Over the last 50 years, there have been several significant outbreaks of this disease. At one epidemiological extreme are areas where West Nile virus circulates in most years and accompanied with West Nile fever is a mild and common childhood age, and West Nile fever epidemics and West Nile meningoencephalitis cases are rare. Based on extensive studies that done in Egypt in 1950s, the ecology of the disease was found to vary greatly depending on the prevalence of the disease. Therefore, high prevalence of background immunity is present and increases with age, and West Nile fever epidemics and West Nile meningoencephalitis cases become rare. At the other extreme are the industrialized urban areas where little or no previous West Nile fever activity have occurred. Aging and immunologically naive populations encounter this virus for the first time, and preparedness of the neuroinvasive cases have been observed. Occupied Palestine had been expressed several outbreaks in 1950 and 1957. Nursing homes reported severe neurological disease and deaths associated with West Nile disease. There have been many West Nile outbreaks throughout the world. 1973 and 1974 it is reported in South Africa. The outbreak in France at 1962 and in Romania at 1969 seemed to spark the beginning of several outbreaks worldwide in large industrialized urban areas. West Nile fever was firstly documented in Western Hemisphere in 1998 in Italy, then in Russia at 1999, and another outbreak in France at the beginning of this millennium. We will discuss in the following the epidemiology of West Nile fever disease. Pathogenic risk factor. West Nile encephalitis virus classification. It is a virus of Realm riboviridae, phylum Incerticides, family Flavoviridae, genus Flavivirus, and the species is the West Nile virus. It is a single-stranded RNA virus. It is a member of the family Flaviviridae, especially from the genus Flavivirus, which also contain Zika virus, dengue virus, and yellow fever virus. West Nile virus developed into two distinct lineages. Lineage number 1 and its multiple profiles in the source of the epidemic transmission in Africa and throughout the world. Lineage number 2 was considered an African zoonosis. However, in 2008, lineage 2, previously only seen in horses in sub-Saharan Africa and Madagascar, began to appear in horses in Europe, where the first known outbreak affected 18 animals in Hungary. Lineage 2 West Nile virus had been detected in equines and humans in South Africa. At 2007, fatal case in a killer whale in Texas broadened the known of host range of West Nile virus to include cetaceans. Lineage 1 West Nile virus was detected in South Africa another time in 2010 in a mare and her aborted fetus. Since the first North American cases in 1999, West Nile virus has been reported throughout the United States, Canada, Mexico, Caribbean, and Central America. There have been human cases and equine cases, with many birds are infected. Both the American and the Israeli strains are marked by high mortality rate in infected avian populations. The presence of dead birds, especially corvidae, can be an early indicator of the arrival of West Nile virus. West Nile virus as appear under electron microscope. West Nile virus is an enveloped virus with icosahedral symmetry, image reconstruction, and cytoelectron microscopy reveal around 45 to 50 nanometer virion covered with a relatively smooth protein shell. This structure is similar to the dengue virus, another flavivirus. The protein shell is made of two structured proteins, glycoprotein E and the small membrane protein M. Protein E has numerous functions, including receptor binding, viral attachment, and entry into the cell for membrane fusion. The outer protein shell is covered by a host-derived lipid membrane. The viral envelope and the flavivirus lipid membrane has been found to contain cholesterol and phosphocetylase, but other elements of the membrane have yet to be identified. The lipid membrane has many roles in viral infections, including acting as a signaling molecule and enhancing entry of the virus to the cell. Cholesterol in particular plays an integral part in West Nile virus entering host cell. The two viral envelope protein E and M are inserted into the membrane. The RNA genome is bound to capsid C protein, which are 105 amino acid residues long, to form the nucleocapsid. The capsid proteins are one of the first proteins created in the infected cell. The capsid protein is a structured protein whose main purpose is package RNA into the developing viruses. The capsid has also been found to prevent apoptosis by affecting the ACT pathways. Genomic composition of West Nile virus. The RNA genome is bound to capsid proteins, which are 105 amino acid residence long, to form nucleocapsid. The capsid proteins are one of the first proteins created in an infected cell. The capsid protein is a structured protein whose main purpose is to package RNA into the developing viruses. The capsid had been found to prevent apoptosis by affecting the ACT pathway. The genome of West Nile virus modified at 2010 and is a positive sense single-stranded RNA virus. It is composed of approximately 11,000 nucleopeptides long and is flanked by 5 and 3 non-coding stem loops. The coding regions of the genome codes for 3 structural proteins and 7 non-structural proteins. Proteins that are not incorporated into the structure of new viruses. The West Nile virus genome is first translated into polyprotein and later cleft by the virus host protease into separate proteins, non-structural C and E. Structural proteins are C, prim M, E are capsid precursor membrane proteins and envelope proteins respectively. The structural proteins are located at the fifth end of the genome and are cleaved into natural protein by proteases. Structural protein C its function act as a capsid protein encloses RNA genome, package RNA into immature virions. The PRM or the precursor membrane protein M, the viruses with M protein are infectious. Presence of M protein allows the activation of proteins involved in the viral entry into the cell. Precursor M or precursor membrane protein is present on immature virions by further cleavage by furin to M protein. The virions become infectious. E protein is glycoprotein that forms the viral envelope, binds to receptors on the host cell surface in order to enter the cell. The non-structural protein consists of non-structural 1, 2A, 2P, 3, 4A, 4B, and 5. These proteins mainly assist the viral replication or act as proteases. The non-structural proteins are located near the 3 end of the genome. Non-structural proteins, all of them are help in finishing the life cycle of the virus. A diagram of genomic structure of West Nile virus, where the E protein, the RNA, capsid protein, and membrane protein, and the above part is speaking about the structure and the non-structure proteins. Phylogenetic tree of West Nile virus based on the complete nucleotide sequences of selected viruses strains, demonstrating genetic relativeness of these strains. Abbreviations denote two different strains. Boxes indicate different lineage and clades. Hungarian strains here reported in this image are highlighted with gray background. West Nile disease distribution. West Nile encephalitis is a worldwide reportable infectious disease that affects equines, mammals, and man. Where the distribution of West Nile encephalitis at early of 2008 shown by this map, where it is present in North America, Africa, parts of Asia, Europe, Middle East, and Australia. Signs that it spread across a contiguous United States. Map elaborated by OIE office at the time of January up to June 2019 denoted that there is no one country reportable of West Nile encephalitis disease. Animal risk factor for West Nile encephalitis disease. Animal risk factors for West Nile encephalitis virus are humans and some non-human primates, horses. One of the viruses with broad host range and also known to be able to infect at least 30 mammalians, dogs and cats. Some infected humans and horses express the disease, but dogs and cats rarely show the symptoms. Reptiles and amphibians can also be infected, including some species of crocodile, alligators, snakes, lizards, and frogs. West Nile encephalitis transmission. This diagram expresses the host range and the transmission and life cycle of West Nile encephalitis virus in the nature. Culex pipiens mosquitoes are vectors for the virus. The natural hosts are birds and mosquitoes. Over 300 different species of birds have been shown to be infected with the virus. Some birds, including American crow, blue jay, and greater sage grouse, are killed by the infection, but others survive, such as American robin, house sparrow, are thought to be among the most important reservoir species in North American and European cities. Brown thrush, gray catbirds, northern cardinal, and northern mock garebird, wood thrush, and the dowie family are among the other common North American birds in which high level of antibodies against the virus have been found. West Nile encephalitis virus have been demonstrated in a large number of mosquito species, but the most significant one for viral transmission are Culex species that feed on birds, including Culex pipiens and Culex salinoris. Experimental infection has also been demonstrated with soft-tick vector, but it is unlikely to be important in natural transmission. Mammals are considered identical or dead-end host for the virus. They don't usually develop high enough level of viruses in the blood to infect another mosquitoes feeding on them and carry on the transmission cycle. Some pairs are also act as a dead-end hosts. In the normal rural or in zoonotic transmission cycle, the virus alternates between the pair reservoir and the mosquito vectors. It can also be transmitted between birds via diet contact, by eating uninfected bird carcasses, or by drinking infected water. Vertical transmission between female and offspring is possible in mosquitoes and might potentially be unimportant in overentering in the urban or spillover cycle. Infected mosquitoes that have fed on infected birds transmit the virus to humans. This requires mosquito species that bite both birds and humans, which are termed bridge vectors. The virus can also rarely be spread through blood transfusion, organ transplantation, or from mother to baby during pregnancy, delivery, or breastfeeding. Unlike in birds, it does not otherwise spread directly between peoples. Transmission. Culex pipiens mosquitoes are a vector for West Nile virus. The natural hosts for West Nile encephalitis virus are birds and mosquitoes. Over 300 different species of birds have been shown to be infected with the virus. Some birds, including American crow, blue jay, greater sage grouse, are likely be the infection. By other survives. The American robin, house sparrow are thought to be among the most important reservoir species in North American and European cities. Brown thrush, gray cartid, northern cardinal, northern mock birds wood thrush, and the doi family are among the other common North American birds in which high level of antibodies against West Nile fever have been found. West Nile fever virus has been demonstrated in a large number of mosquito species, but the most significant for viral transmission are Culex species that feed on birds, including Culex peepons. Experimental infection has also been demonstrated with soft tick vectors, but it unlikely be an important in natural transmission. This diagram describes the West Nile transmission cycle where the eggs of the male and female mosquitoes can carry the virus for the fifth generation and then give them to end hosts as birds, as squirrel and rats and equines, and from them it can be transmitted through mosquito also to humans. So we have dead end hosts as the equine and human. This diagram clarifies how one mosquito can take the infection from a host such as birds or squirrel or the other 30 mammalians, and then that enter the virus into another host. Then another mosquito take strong host purifying selection of the viruses, and another one will transmit to another host. So the virus, if it's coming from one specific host to the same specific host, it becomes strong and make the infection. T here is reported to the transmission number of the virus. Sources of infection. Culex species are the most important vector in maintaining the West Nile encephalitis virus cycle. However, 43 different species of mosquitoes have been found harboring this virus in the United States. Many different types of mosquitoes throughout the world have also been found to transmit this virus. West Nile encephalitis virus has been isolated from ticks in Asia and Russia, but their role in virus transmission is still unclear at this time. Upper photo shows mosquitoes larvae in the water. Lower photos shows female Culex mosquito laying eggs on water is an egg raft formation, and this is clarify the transmission of the virus from one generation to another in the mosquitoes. The exact mechanism of the presence of West Nile virus in the environment is not exactly known. However, recent studies have demonstrated several possible mechanisms which may work in contact to provide the distribution picture seen. The presence of overwriting Culex mosquitoes has found in New York City following the initial discovery in 1999. A study in India confirmed that transovarial transmission does occur in Culex mosquitoes in low rates in laboratory setting. Studies completed in birds also indicated that contact transmission between them may occur, and migratory birds may have large role in transporting this viral and its vector to unaffected regions. Molecular pathogenesis. Once West Nile encephalitis virus has successfully entered the bloodstream of a host animal through different species of mosquitoes or hematogenous insects, the envelope protein E binds to attachment factor called glycosaminoglycans on the host cell. These attachment factors aid entry into the cell. However, binding to primary receptors is also necessary. Primary receptors including DC sign R, and the integrin alpha and beta. By binding to these primary receptors, West Nile virus enter the cell through clarithin-mediated endocytosis. As a result of endocytosis, West Nile virus enter the cell within an endosome. The acidity of the endosome catalyzes the fusion of endosomal and viral membranes, allowing the genome to be released into the cytoplasm. Translation of the positive sense single-stranded RNA occurs at the endoplasmic reticulum. The RNA is translated into a polyprotein, which is then cleaved by viral proteases, non-structure 2B and non-structure 23, to produce mature proteins. In order to replicate its genome, non-structure 5 RNA polymerase forms a replication complex with other non-structure proteins to produce an intermediary negative sense single-stranded RNA. Negative sense stranded serves as a template for synthesis of the final positive sense RNA. Once the positive sense RNA has been synthesized, the capsid protein C encloses the RNA strands into immature virions. The rest of the virus is assembled along the endoplasmic reticulum and through the Golgi apparatus, and results in non-structural infectious immature virions. The E protein is then glycosylated and BRM is cleaved by furin, a host cell protease, into the M protein, thereby producing an infectious mature virion. The mature viruses are then secreted out of the cell. This is a schematic diagram of West Nile encephalitis virus multiplication cycle. Here, at the left part of the picture, upper level, the virion enter and then receptor mediated and cytosis, then uncoating of the virus, and the different processes we explain in the molecular pathogenesis occur, where here a vesicle come with progeny of another multi-hundreds of virions come out. This will cause the destruction of the part of the brain that affect with it. This picture explains the process of meningoencephalitis, where after the blood stream come loaded with the West Nile virus and the multiplication, as we said in the molecular pathogenesis, occur in the neurons of the brain, destruction of certain centers in the brain will giving the clinical symptoms, such as centers of mechanism. If it's affected, it will give paralysis and so on. Now, see you all in the next video for explanation, the second part of West Nile encephalitis infectious disease.